Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Simplifying Assay Development with Molecular Standards, Remove Culturing from the Equation, presented by Mr. Kyle Young. Mr. Young is a product line business specialist and scientist at ATCC. In this presentation, Mr. Young will provide an overview on the current challenges in the development of molecular-based assays. A special focus will be placed on ATCC's wide range of molecular standards, from human pathogens to microbiome components. Mr. Young will then provide application data demonstrating the potential use of these products in diagnostic assay development. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Kyle Young.
Thank you, Brian, and thank you to everyone for joining us today. This webinar is broadly about ATCC's molecular standards, so I'll start with a description of what they are and then talk through the development process of a molecular diagnostic assay to show how they can be used to speed up that process. I'll also do a species selection exercise and show you how to quickly find a molecular standard on the ATCC website. Next, I'll cover the development process for the synthetic standards. And last, I'll talk through some of the internal validation work that we've done. But first, some background on ATCC. The American Type Culture Collection was founded in 1925 and is a private, independent, nonprofit organization headquartered in Manassas, Virginia, with an R&D facility in Gaithersburg, Maryland. ATCC is the world's premier biological materials resource and standards development organization. We collaborate with and support the scientific community with industry standard products, innovative solutions, and a growing database of bioinformation. And we have an enormous range of materials in our collection that includes cell lines, organoids, microorganisms, various derivatives such as GFP or luciferase tagged organisms, nucleic acids, knockout cells, and high titer viruses. We have NGS standards, a curated collection of antimicrobial resistant organisms, media and reagents, and the focus of today's talk, the molecular standards. Each of our biomaterials are maintained at low passage and are authenticated and characterized using genotypic, phenotypic, and/or functional assays so that you have a credible starting point for your research. Let's begin with a brief explanation of the molecular standards and a sample case of how they can be used. ATCC's molecular standards are DNA or RNA preparations that are quantified by ddPCR, are backed by ATCC's extensive quality control protocols, and are downgraded to BSL-1 with a few exceptions. The quantitative format is primarily what differentiates them from the other nucleic acid preparations in the catalog. We have two basic types: synthetic standards, which are de novo constructs of DNA or RNA, and genomic standards, which are extracted from genuine ATCC cultures. The synthetics make possible standards for organisms that are difficult to culture, either because the organism doesn't grow in vitro or because it requires a high biosafety level containment. They also make it possible to work with a higher concentration of target DNA for organisms where DNA extraction is difficult, such as Ureaplasma ureolyticum. They don't include a full genome, but rather focus on a few highly relevant gene sequences. These were born of ATCC's desire to be more than a culture collection and to continue to improve and expand on the resources that we provide. On the other side of the slide, the genomic standards are extracted directly from ATCC cultures and contain the full organism genome. A new benefit for the genomic standards is the ATCC Genome Portal, where full sequence data for ATCC cultures are available and searchable for free. Since we launched it in 2019, we've sequenced almost 1,300 organisms, including sequences for about 65% of the genomic molecular standards. This is an ongoing project, so that number will grow over time as we continue to work on improving the value of our materials. The specifications for the two types are similar. The synthetics are fully sequenced to verify the construct matches our design, and we test them using at least one published assay, which we report on our website. The genomic standards are authenticated by amplicon sequencing of a relevant gene target for the specific organism, and the nucleic acid integrity is verified by gel electrophoresis, except for the viruses. Each standard type is filled with 100 microliters at 10 to the 5 to 10 to the 6 copies per microliter, with the specific construct or genome count reported on the certificate of analysis. Both types are shipped frozen with storage recommendations based on the nucleic acid type, either negative 70 degrees Celsius for RNA or negative 20 degrees Celsius for DNA. The molecular standards are of primary utility in the development, verification, validation, or control of a molecular diagnostic assay, one that employs next-generation sequencing, or more commonly, a nucleic acid amplification test, like PCR or an isothermal protocol. The slide I'm showing now is a diagram of some of the major points on the development side of a molecular diagnostics assay. Once the target organisms and basic type of assay to be used are identified, the two major categories of design and experimentation are for sensitivity and specificity. Assay sensitivity refers to inclusivity testing and limit of detection testing, which are different ways of evaluating the chance the assay would return a false negative result, or a negative assay result when the target organism is present. Inclusivity simply means that you're testing to verify that your assay detects what it is supposed to detect. So, does my assay for respiratory pathogens give a positive result for influenza A H1N1? What about for H3N2, or influenza B, or respiratory syncytial virus? What about different isolates or strains or lineages within those categories? It's important to be thorough and test multiple strains of the target. So this line of testing can be very extensive for multiplexed assays or for assays that are designed to detect highly variable pathogens, like a virus with an RNA genome. In some cases, the breadth of testing can be mitigated with in silico analysis, but a certain minimum of bench work is going to be required at some point. These assays can also entail a great variety of organisms, like for a broad respiratory pathogen test, and it can be a difficult long-term burden to gather the expertise and equipment needed to grow, extract, and quantify those organisms in-house. Even if that work is outsourced, there are still risks to project timelines because organisms take time to grow, and some, like Mycobacterium tuberculosis, can be very, very slow. And that's why many labs choose to use an off-the-shelf solution, like ATCC's molecular standards. They save both time and money by removing a layer of complexity from the project. Here's a look at a sampling of our menu of pathogens. From the first 4 standards launched in 2013, the molecular standards have grown to 236 in the first half of 2021, 173 genomic and 63 synthetic. So there's too many to fit on a single slide. This just gives an idea of the different pathogen types where we can support an assay design or research project. And so don't worry about reading the list. I'm going to come back to this slide a couple of times. We've done a lot of work making standards for pathogens of all types, including bloodborne pathogens such as HIV and hepatitis C, gastrointestinal pathogens like astrovirus, cryptosporidium parvum, and Salmonella enterica, respiratory pathogens like the coronaviruses and influenza viruses, sexually transmitted infections such as human papillomavirus, herpes viruses, and chlamydia trachomatis, vector-borne pathogens like dengue virus and West Nile virus, and opportunistic pathogens like Staphylococcus aureus and Candida albicans. The selection process of strains for inclusivity testing depends on the ultimate purpose of the assay and its scope. It's important to make a clear decision on your design goals early on. Here, I'm showing some pathogens that are commonly cited for surveillance of water and food safety. There's often crossover between these lists because both groups are gastrointestinal pathogens. If you aren't clear what your assay needs to do, you'll wind up with scope creep. So just a basic reminder to design your experiment first, and then go to the lab. There's also a bit of project history on this slide, in that it covers 3 of the first 4 molecular standards at ATCC. Pictured at top right is catalog 29212Q-FZ. This was a kit for Enterococcus faecalis strain Portland, cited by the EPA for water quality testing. This was made as a panel with serial dilutions and reagents included. It has since been simplified into a single vial with a DQ suffix, like the rest of the genomic standards. At bottom right is an image of norovirus, and constructs for genogroup 1 and 2 were the first synthetic standards that we launched, along with a construct for West Nile virus. I'll come back around to speak more on the noroviruses later in the presentation. One particular pathogen that's on most people's radar is severe acute respiratory syndrome-related coronavirus 2, and that's where I'll focus the assay design exercise. I'll start with some explanation of the catalog numbers we're looking at here. Starting with the list of synthetics on the left. At the beginning of the pandemic, ATCC was focused on synthetics because that was the fastest tool that we could get to the catalog. Our constructs here are focused on the assay targets as determined by various governing bodies, most notably the CDC, the Chinese CDC, and the WHO. You'll notice that each of the constructs is described with a list of genes. These are the genes that are included on that construct. As clinical isolates became available, we evaluated the risk of working with and distributing these and determined that only BSL downgraded derivatives would be in the ATCC catalog. This kept our risk low while still making materials available to most labs. The list to the right are the genomic RNA preparations that are available or expected to launch in the near term. Because SARS-CoV-2 has a positive sense RNA genome, we're only able to downgrade these to BSL-2. The first isolate to the catalog was the reference strain for North America, Washington 1, 2020, first in genomic and then in heat and activated format. We tried to diversify based on geographical isolation at first, and now that the phylogeny has been formalized, we're working on sourcing the CDC variants of concern and variants of interest. No variants of high consequence have been identified yet, but we would prioritize any of those that are designated as such. In addition, we're trying to source these lineages in the United States because CDC permits are now required for shipment of internationally sourced materials. So for our thought experiment, let's say that we want to design a multiplex quantitative RT-PCR assay for SARS-CoV-2, influenza A, influenza B, and respiratory syncytial virus. Let's further say that our target for the SARS-CoV-2 portion of the assay is the RNA-dependent RNA polymerase gene. We wouldn't be interested in VR3277SD or VR3278SD for inclusivity because the spike gene is out of scope for the assay. But VR3276SD and VR3279SD would be worthwhile to include because these could be suitable as assay controls later on. We'd also want to test all of the genomic preparations for SARS-CoV-2. Returning to the sample list of organisms, we also want our inclusivity assay to cover several strains of RSV, different lineages of influenza B, and different serotypes of influenza A. Once you've established that you're detecting the proper targets, it's important to determine the lower limit of detection. This typically involves choosing representative members of the inclusivity panel and then performing a series of endpoint serial dilution experiments in a real or contrived sample collection matrix. This could be viral transport media, synthetic saliva, or some other collection buffer. A standard curve may also need to be generated for some assay types, like qPCR, and there may also be tests to establish ratios of genome copies with a secondary calibrator, like plaque or colony-forming units, or tissue culture infectious dose. Whatever the scope of work, accurate quantitation is very important because LOD results are reported to regulatory bodies and to customers. Practically, you want to achieve a limit that's as low as possible because a low limit of detection gives your end users confidence in the assay. And whatever load of the pathogen is detected, it's useful to know that it's there. This is especially true of environmental surveillance assays, like those that test for SARS-CoV-2 in wastewater, because there are epidemiological ramifications to finding even trace amounts of viral RNA. For our hypothetical assay, we want to choose reference strains or well-documented isolates for each virus species. For SARS-CoV-2, that would be the Washington 1 isolate, catalog VR1986D. We might also choose catalog VR3276SD as it's a good candidate for a control material, being slightly less costly and BSL-1. We'd also identify at least one isolate from each RSV subtype, each influenza B lineage, and each influenza A serotype from within our assay scope. The other major thrust of testing is for assay specificity or exclusivity testing. You may also hear it called cross-reactivity testing. In these experiments, an assay is tested for potential false positives, falsely reporting the target is present when it is not. This involves running a whole host of other organisms through the assay to make sure that it doesn't react with any other organisms that could potentially be in a test sample. Strain selection here is relative, so microbiome-associated organisms and human cells are often implicated, but Bordetella pertussis could be used for the inclusivity panel of a respiratory assay and for the exclusivity panel for an enteric assay, and vice versa for E. coli. Exclusivity tests often range even more broadly than an inclusivity panel. A recent example is the FDA's recommended list of organisms to be analyzed for SARS-CoV-2 diagnostics. Their list contains 24 groups of organisms that are very broad in several cases. For example, influenza A and B are grouped together on a single line in the recommendation letter. But functionally, many assay developers used numerous different isolates in their testing. The same is true for adenoviruses, enteroviruses, parainfluenza viruses, and others on the list. For the first generation of diagnostics targeting SARS-CoV-2, all other organisms on the list were intended for exclusivity testing. In an example of the inclusivity-exclusivity swapping I mentioned on the previous slide, the RSV and flu viruses became part of the inclusivity panels for many of the second generation of SARS-CoV-2 tests, as we're doing for our thought exercise. I've highlighted the rows where we've identified species for inclusivity and limited detection testing in our assay, and so the rest of these organisms would be the minimum for our exclusivity panel. I say minimum because we would also probably want to choose to test a few more microbiome-associated organisms. If you look at this list, the coronaviruses, the adenoviruses, rhinoviruses, TB, Chlamydophila pneumoniae, Streptococcus pneumoniae, Bordetella, it's very heavily focused on respiratory pathogens. And if you're going to undertake all of the work to make an assay, it's nice to know that it won't be derailed by a stray E. coli or Cutibacterium acnes in a patient sample. Mapping out the list in our menu reinforces the basic idea behind exclusivity testing. If we're running a respiratory diagnostic test on a patient, it's because they're experiencing respiratory symptoms. Our assay is being used as a tool to help the physician either identify or rule out some of the more serious possibilities. So there's a possibility that a rhinovirus or an adenovirus is causing a severe cold, and we definitely don't want to lead the physician to misdiagnose this as COVID-19. One last important point on inclusivity versus exclusivity testing. While inclusivity testing examines for a specific binding target, exclusivity testing is looking for any binding target and requires a full genome of the assayed organisms. Because synthetic products often don't include a full genome, they're not usually suitable for exclusivity testing. With design goals and an experimental plan in hand, we're ready to start lab work. The easiest way to find what you need on the ATCC website is the search bar. You can also start with the drop-down for your area of focus. For our test, it's virology. For each collection area, you'll see that nucleic acids is an option. At the virology nucleic acids page, you can scroll down to the bottom and reach the product listing. Many of these products have qualitative release specifications. To see just the quantitative options, click the box for molecular standards to filter the results. You can also filter results by organism, genomic versus synthetic standards, and nucleic acid type. And if you can't find what you want, please let us know. You can send an email to me directly at kyoung, K-Y-O-U-N-G, at atcc.org, or to our technical services team via the website. We're still growing this catalog group, and your input would be very helpful. In 2018 and 2019, we did a lot of work internally to build up the process for making genomic viral nucleic acid standards. Many of those had very timely releases to the catalog in 2020 when there was a great deal of demand. Our current work is primarily focused on quality control strains, those cited by a pharmacopoeia, the Clinical Laboratory Standards Institute, or another recognized authority. We're also continuing to make new pathogen preparations to support different areas of development. What comes next after that will be based on your feedback. ATCC exists to be a resource for science, so we want to know what it is that you need. What would make your work easier, faster, less costly? Please let us know. Okay, let's say that we have developed an assay. The next step is validation, to make sure it works as designed and is properly documented. Let's leave our design exercise behind and look at some real data. These were generated for our human herpes virus 1 and 2 genomic standards. We set up standard curves shown in blue for HSV1 on the upper panels and HSV2 on the lower panels. And these range from 2 times 10 to the 5 copies to 2 copies by serial 1 to 10 dilution. Viral DNA from the National Institute for Biological Standards and Control, HSV1 and HSV2 working reagents, was extracted using a commercially available kit. And these samples are shown in red on the amplification curves. These qPCRs were used to quantify the genome copies in the samples within a very narrow interval near 10 to the 2 genome copies per microliter. We ran this assay several times to confirm results. Consistency at this step is paramount because these experiments won't be yours alone. Your end users will be running them as well as part of the performance qualification in their lab. Finally, assays need regular controls to monitor run-to-run and lot-to-lot variation. Here, we sent HSV material to the Cincinnati Children's Hospital Medical Center for trial use. The DNA was diluted 1 to 1,000 and used in a laboratory-developed test in the pathology department over a 3-month period by 3 different operators in 36 assays for each virus. These data were tracked and are graphed by the threshold cycle count for each run. Overall, the consistency is very tight, with a single outlier. During the test period, the HSV1 control had a standard deviation of cycle count threshold of 1.14, and the HSV2 control had a standard deviation of 0.54. That covers a very high-level overview of the development process. There's a lot more to learn, and if you're interested in doing some reading, the FDA has compiled a list of all the emergency use authorized assays developed during the SARS-CoV-2 pandemic. You can find them on the FDA website by searching for In Vitro Diagnostics EUAs, and you'll have dozens and dozens of development case studies where you can learn more about these and the other steps in the process, like sample collection, processing, and extraction protocols. Next, I'm going to change gears a bit and go into a little bit more depth on the design process for the synthetic standards. And I'll start with some improvements we made from the first generation synthetic constructs, which included a West Nile virus and norovirus genogroup 1 and 2. These were designed by comparing targets from published assays, diluted to a target concentration in TE buffer, and then delivered in a dried format. While the initial interest in the standards was high, we began to find some common threads in our feedback that there were some specific points that could be better. So armed with that constructive feedback, the norovirus constructs were redesigned, and the R&D team presented the changes in a poster at the Clinical Virology Symposium in 2015. First, the preservation format changed from dried to frozen, and an RNA stabilizer was added. These first 2 changes increased shelf life dramatically. The quantitation method was changed to droplet digital PCR for a more accurate copy count. Manufacturing with ISO 13485 guidance was adopted for laboratory operations. And last, the construct itself was expanded by adding a portion of the RNA-dependent RNA polymerase gene to increase the number of published primer sets that would bind. The next several slides are from the poster that we presented at CVS, titled Development of Improved Synthetic Molecular Standards for Norovirus Genogroups 1 and 2. The full poster is available on the ATCC resources page for posters presented in 2015. First, the performance of the new constructs. We chose 2 third-party primer and probe sets, one from CaliciNet, CDC's norovirus outbreak surveillance network, and another recommended by the European Committee for Standards. This slide shows the standard curves using the CaliciNet assay for the norovirus G1 in the upper panels and norovirus G2 construct in the lower panels. For both new constructs, we have a nice linear progression between dilutions. We ran the ECS assay as well, with similarly good results, reported in the box at right. With two sets of constructs, the obvious next step is to compare them. Here we have a direct comparison of the old and new Genogroup 1 constructs using the CaliciNet assay and ECS assays, with the new construct in blue and the old in green. You can see on the upper panels that the new construct performs similarly to the old using the CaliciNet assay. However, we show on the lower panels that it dramatically outperforms the old one when using the ECS assay, where the primers didn't bind well at all to the first construct. Standard curves for the Genogroup 2 construct were also compared, again shown with the CaliciNet assay in the upper panels and the ECS assay in the lower panels. And the new construct shows a slight leftward shift in the amplification curves for both, indicating that it's a bit more efficient than the old construct for both assays. Last, we used the respective standard curves to quantify the working reagents for norovirus genogroups 1 and 2 from the National Institute for Biological Standards and Control. Here, sample amplification plots are shown with the standard curves in blue and the NIBSC working reagents in red. The working reagents were assayed in triplicate using each assay at undiluted, 1 to 10, and 1 to 100 dilutions. Our results showed that there were 2.3 times 10 to the third genome copies per microliter in the norovirus 1 reagent, and 2.4 times 10 to the 2 genome copies per microliter in the norovirus 2 reagent. Gathering all that we learned from our initial design and then the redesign, we constructed the following basic process for making synthetic standards. First, we vet the need for the construct based on public health concern, volume of published research, and direct feedback from the research and diagnostic communities. Then, we collect as much relevant data as we can from public sources in order to assemble a draft construct. Our designer conducts a series of literature and bioinformatics analyses to determine the most valuable genetic fragments to include. After that, we have one last technical review to finalize the construct. Following completion of the design, we have a regulatory review to make sure that we don't make anything that could be potentially dangerous, like a full toxin gene. And then the construct is actually produced, assays are double-checked and optimized, and last, vials are filled and tested. There are a number of questions that we commonly field at conferences and through our technical service department, and before I move on, I'd like to answer a few of them. The synthetic standards are small, typically less than 5,000 nucleotides in length. Because of their size, in most cases, they do not include entire genes, only gene fragments. There's enough sequence there for assays to function, but the amplicon size may not always match what you'd find from a wild-type genome. Our choice of nucleic acid is made to match the natural genome, so there are several RNA constructs, like the norovirus standards that we already discussed. We recognize that you need a starting point for your work, so we do report an assay that we've confirmed to function on the website. Other assays can be determined by looking at the gene fragments included in the construct or at the references on the website. The sequence is proprietary, so we won't disclose it, but we're happy to confirm whether your primers and probe will bind to it prior to a purchase. In the last segment of the presentation, I'll share some work ATCC did in support of a poster that we brought to the Clinical Virology Symposium on 2019 for hepatitis viruses. Just a quick note first, hepatitis viruses are named for the disease they cause based on infection of the liver. Other than that, they don't necessarily have much in common, including their classification, which places them in different families of viruses. For example, hepatitis B is a member of Hepadnaviridae and has a DNA genome. Hepatitis C is a member of the Flaviviridae and has an RNA genome. This project was similar in many ways to the norovirus redesign analysis. First, we generated a standard curve using our hepatitis B and C virus standards. On this slide, you see the hepatitis B qPCR curve, which ranges from 3 times 10 to the 5 copies to 3 copies in serial 1 to 10 dilutions. The NIBSC hepatitis B working reagent was reconstituted in 500 microliters of molecular grade water and extracted using a commercially available kit. Only one curve is shown, but this assay was performed 3 times, with each sample run in triplicate for each, demonstrating strong linearity. We did the same for the hepatitis C standard. The standard curve for qRT-PCR was set for 10 to the 5 copies to 10 copies by serial 1 to 10 dilution. The NIBSC hepatitis C working reagent was again reconstituted in 500 microliters of molecular grade water and extracted using the same commercial kit. Each dilution was run in triplicate, and the assay was repeated 3 times again with very strong linearity. For this project, we also ran ddPCR. This slide shows the one-dimensional amplitude scatter plots of positive and negative ddPCR for 3 dilutions from 10 to the 4 to 10 to the 6 in triplicate for HBV on the left and hepatitis C on the right. This assay used the same primers as the qPCR and qRT-PCR assays that I showed in the previous 2 slides. And here we're also seeing a consistent progression between dilutions. Because the NIBSC reports infectious units per milliliter, we were also able to calculate the number of genome copies in an infectious unit as being 11.4 genome copies per IU for the hepatitis B reagent and 61.5 genome copies per IU for the hepatitis C reagent. The data that I've shown today was pulled from posters that ATCC presented at the Clinical Virology Symposium in 2015, 2016, and 2019. We've also presented posters for our dengue virus standards in 2014, our Zika virus standard in 2017, and our human parvovirus 3 standard in 2018. There's much more additional information at the ATCC website in our new resources section, where you can find posters, presentations, culturing guides, white papers, and more. So I realized that I went pretty fast through some of the data slides, and if you'd like to go through those at your own pace, you can access all of the information there. In summary, ATCC molecular standards are a consistent, reliable starting material for assay development and control that eliminate the cost of growth, extraction, and quantitation. ATCC has over 230 total standards, including over 60 synthetic constructs, that cover many common organisms with human health implications, and we're continuing to make more of these. Last, I'd like to thank everyone who worked on this webinar, either by generating the data, creating the standards, or producing this event. So thank you to our R&D group, the technical transfer, and marketing teams, both past and present. I'd also like to give a very warm thank you to our outside partners, to Donna Diorio at Cincinnati Children's Hospital, to the National Institute for Biological Standards and Control in the UK, and to Dr. Benjamin Pinsky at Stanford for beta testing our early synthetic standards. All right, are there any questions?
Well, thank you, Kyle. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. All right, so let's go ahead and jump into our first question. So Kyle, how are the genomic standards extracted?
So we try to use a commercially available kit where possible, but that doesn't always yield acceptable results for every organism, so we have to use classical methods for some cultures. Okay.
So another question that's come in is, why is 29212Q-FZ being reworked?
So basically, that product was our first attempt at a molecular standard product, and it was kind of complicated and very expensive. So it had come with several dilutions of, that were prepackaged at specific concentrations for a set dilution experiment, and it had some other reagents that were packaged in with it, and so the cost was not reasonable, and so we simplified it to align with the other genomic standards and reduce the price.
Okay, good. All right, this next question, actually, this is an amalgamation of a couple questions that have come in, but can ATCC prepare custom standards?
Yes, we can. So you can order any kind of custom work at all by contacting our custom services team on the form in the services page of the website, and just let them know what you want, and we'll work up a quote for you.
All right, great. This is a question that actually I had up until recently, but this might be helpful for the audience. So what's a DQ product? And then also, what is an SD product?
Right. So at ATCC, because the collection is so large, we try to maintain the base catalog number for all of the cultures, and then derivatives will usually get either a prefix to that or a suffix to that. So the DQs is the suffix that we put on a molecular the molecular version of the live culture. So that's DNA in a quantitative format. And then the SD is a synthetic DNA. I guess come up with that. We didn't want to confuse everyone by adding RNA in there, so they're both DQ or SD. And then for the SD products, there's no base culture associated with that item number, just to make sure that we don't confuse people, because there are some synthetic products that are covered by the United States Commerce Control List that can't ship internationally.
Okay. All right, good. Interestingly, that's the next question. Is there any global shipping restrictions, I guess generally speaking, to the molecular standards?
So as I mentioned, some of the synthetic constructs in the most recent update to the U.S. Commerce Control List, that covers now genetic materials. So even if it is synthetically derived, if it is imitating, say, I think West Nile is on the list, or certainly all of the the select agents as described by the U.S. Patriot Act. Those are all on the Commerce Control List. ATCC does not distribute those internationally. In addition, there are some of our distributors that aren't able to import the materials. So, for example, we very recently got approval to ship heat inactivated SARS-CoV-2 to Australia. So yes, there are some, and if you are working with a distributor, if you just contact them, they'll be able to find out for you what they're able to get.
Okay, good. All right. There's actually a follow-up question where the customer is asking if they can become a distributor, and we would probably need to be following up with them directly. All right. Let's see. Oh, here's a good question. Can we calculate the number of bacteria by having the genomic copy numbers in the standards?
We calculate the number of bacteria by having the genomic copy numbers in the standards. I think, you could establish a ratio if you were running experiments in your lab. There are going to be variations in extraction efficiency. So whatever assay that you're working with, you'll have to do an additional set of experience to figure that out and make sure that you're normalizing your data correctly. But yes, the molecular standards are coming with an accurate absolute quantitation by ddPCR, so they are a good starting point for those experiments.
All right, good. Now, this question's around the shelf life of the standards. Why are standards and controls, why do they have a very limited shelf life? But they're stored in ultra-low temperature conditions.
So I'm looking at the certificate of analysis for a couple of the genomic standards, and they don't have an expiration date assigned to them. The synthetics was based on, I think, our experience with that first round of synthetics that we made, because there were some stability issues with those because they were in a dried format. So we added a biostabilizer that was recently pulled off the market. So we are we're working on the R&D for our own biostabilizer now, and we're going to have to run all of those stability experiments. If you store those at the generally accepted temperatures, though, there may still be utility after the expiration date, but we would urge you to retest that and consider the risks of doing that.
Gotcha. So that kind of answers this next question. Have you tested stability, right?
Yes. So for the genomic, because we store those at the like the generally accepted temperatures, we're very confident that those are stable. We don't have a regular stability program, however. For the synthetics, we know one of the problems with the first couple that we made was that they were very small and there were no padding on the 5' and 3' ends of the construct. So we've taken some steps, and we are going to be getting the stability data going forward and have a much more comprehensive answer for that in the future. But when they're stored at the proper temperature, they are stable.
All right, good. Okay, well, if we don't have any more questions come in, at this time we will conclude the Q&A session. So thank you, Kyle, again for the excellent presentation, and thank you everyone for attending the webinar. Please join us for more webinars in the ATCC Excellence in Research webinar series. So on July 1st, Dr. Robert Molestina will present his talk on developing molecular approaches to detect babesiosis. And on July 22nd, I personally will give my talk on how you can expand your cell-based assays with an unlimited biologically relevant resource. Thank you again, everyone, and have a great day.